Alright, this video is a continuation of Learning Targets 13 and 14, and we'll be talking about the Krebs cycle and the electron transport chain. And we left off talking about fermentation, and fermentation occurs if oxygen is not present in the cell. Well, if oxygen is present in the cell, the pyruvate that is made in glycolysis travels to the mitochondria. Here's a picture of mitochondria, and mitochondria have several distinct features, as you can tell here. Mitochondria have two membranes. One is this darker brown membrane that I've outlined in purple here, and the other one is this inner membrane that has lots of folds. And these folds, which are called cristae, provide the mitochondria with lots of surface area in order to do lots of work on the cell or on the membrane. The inner membrane is where the electron transport chain is located, and this inner membrane space, the space between the two membranes, is where a concentration gradient will be built up during the electron transport chain, which we'll discuss momentarily. And then this space inside the inside membrane is called the matrix. And the matrix is where the Krebs cycle takes place. I have labeled part of this the pre-Krebs cycle, and only because there is something that it has to occur in order for the Krebs cycle, or in order for to have a molecule to get into the Krebs cycle, but we'll still consider this part of the overall Krebs cycle. And again, we're starting with our molecule pyruvate. And it's important to note that were two pyruvates made, because if you'll remember, we had a glucose that was C6, and we made two pyruvates that were each C3, and so we're dealing with just one of those now going through the Krebs cycle here, and the other one also will go through the Krebs cycle. So as we make some totals here of the Krebs cycle, and as we go through this, recognize that for each glucose molecule, this cycle is going to be happening twice. So pyruvate begins its journey into the Krebs cycle, and the first thing that it's going to do is it's going to lose a carbon. And each time a carbon is lost in this cycle, that carbon is lost as a CO2. And also, each time a carbon is lost, if you can think about it, the bond between one carbon and another carbon is broken, and when that bond is broken, energy is released, and energy is released in the form of electrons. Well, those loose electrons are going to be picked up by our electron carriers. And so here you have NAD plus that comes in and picks up those electrons and turns to NADH. And NADH is now headed off to the electron transport chain, along with all of the other electron carriers. If we create an electron carrier in this process, it's always headed to the electron transport chain. And so at this point, we have two carbons left, and this molecule is going to be called acetyl. And that's going to help us when we understand what's going to happen next. Well, acetyl needs some help to jump onto the Calvin cycle. For some reason, I've chosen this as my CoA color. Well, acetyl is going to get some help from an enzyme called coenzyme A, or CoA. And when CoA picks up acetyl, this molecule becomes acetyl CoA. So again, remember, enzymes take two things and combine them. It's a way of speeding up the reaction. And so acetyl CoA now, which is the coenzyme A plus the acetyl, is what's actually going to be entering in to the Krebs cycle. It's like jumping on the merry-go-round here. Now, acetyl-CoA is going to join up with a 4-carbon molecule, which for our purposes will remain nameless, thankfully, and we'll keep that that way throughout this cycle. We're just going to call these molecules by the number of carbons that they have. And acetyl, a 2-carbon molecule, is going to join to this 4-carbon molecule, and that, of course, is going to create a 6-carbon molecule. And this one we will give a name because the name of this molecule is another name of this cycle. So this is called citric acid, and this is often called the citric acid cycle. So the 2-carbon acetyl is joined to the 4-carbon molecule with the help of coenzyme A. Now coenzyme A is now free to go back and pick up some more acetyl, which is its lot in life. All right, so we have 6 carbons, 2 of which were added to the party, and we need to end up with 4. And so those 2 carbons that are extra, we're going to lose. And again, every time we lose a carbon in this cycle, we're going to lose it as a CO2. And every time a carbon is broken off and those bonds are released, energy is released, the bonds are broken, and electrons are going to be freed up. And every time electrons are freed up, we have our trusty electron carriers that swoop in and save the day. And so what do we have now? We have 5 carbons. Well, we're going to lose one again. CO2, same thing happens, just like you'd expect. Bonds broken, electrons are freed. NAD plus comes in, grabs them, grabs those 2 electrons up, turns to NADH. Now, something else happens here. There's a phosphate that is available when this 5th carbon is broken off, and that phosphate is almost like it's just ripe for the picking. And so an ADP is going to come in and pick that up and turn to ATP. And again, this is not a chemistry class, so we're going to word things like that. If you want to know all the chemistry, I'd be glad to show you some other time. But for our purposes, phosphate is there and is picked up and turns to an ATP. And we now have 4 carbons left. Well, these 4 carbons here are not the same as the 4 carbons that we started with. They have the same number of carbons, but the ones that we started with are in a different shape. And in order to bend things and get shapes, we have to break bonds and reform bonds. Well, again, by the breaking of these bonds, and these are enzyme reactions, so enzymes are actually kind of fitting these, almost like a transformer toy being turned from a T-Rex to a man or whatever. And we have these bonds are being reformed and broken, and electrons are freed up. And so NAD plus is going to pick up some of those electrons, turn to NADH, and then we have the Saturday mail carrier of the electron carrier world, FAD, who only comes around once every so often. And FADH2 is what he turns into. Again, FADH2 is no different in its purpose than NADH. It is still an electron carrier, so don't get confused here. There's just a different electron carrier with a different name. Now, all of these electron carriers are headed off to the electron transport chain. And if you remember, what was the purpose of the Krebs cycle? The purpose of the Krebs cycle was to take pyruvate and to break it down and to make electron carriers like NADH and FADH2, and we happen to get an ATP to boot. So before we draw the electron transport chain, let's take a moment to talk about the totals that we have in the Krebs cycle. So for one pyruvate, we have one ATP that is created, 4 NADH that are created, and 1 FADH2. The 3 NADH in the actual cycle and 1 in the pre-Krebs cycle that we mentioned. Well, what does that mean for a glucose molecule then? Because remember we said how many pyruvates are in a glucose? 2. And so for every glucose that's broken down, all of these numbers are going to be multiplied by 2. So for every glucose, we have 2 ATP. We have 8 NADH, not P, and 2 FADH2. And all of these molecules are headed to the electron transport chain, which I will begin drawing now. So in our electron transport chain, what I'm drawing here is the inner membrane of the mitochondria. Remember, the mitochondria has 2 membranes, and on this membrane there are several proton pumps. I bet I could make more room in between my proton pumps. And these proton pumps do exactly what their name suggests. And I'm drawing an arrow on them because this is the way they pump the arrows. And once again, making a starring role in a body or in a organism process is our friend ATP synthase, which this might be the worst drawn ATP synthase of all time. So this is the inner membrane of the mitochondria. Just orient us here. Above is the intermembrane space. And so below would be the matrix where the Krebs cycle has just taken place. And so our electron carriers are headed to the electron transport chain. And so the first one we're going to deal with is our trusty ally, NADH. Now NADH has 2 electrons, and NADH is going to drop them off at one of these proton pumps. It's going to turn back to NAD plus, and it's going to head to the Krebs cycle to pick up more. Now, those 2 electrons are going to travel through these proton pumps. The proton pumps are basically going to be handing these electrons to each other. The first one's going to take it, get energized, hand them off to the second one. The second one's going to get energized, hand it off to the third one. And by the time the third proton pump has taken and used this electron's energy, this electron is tired. And we'll talk about what happens to this tired electron. But what happens with these proton pumps, because of the energy of the electrons, protons are going to be pumped across the membrane. Through these pumps. And what are we building up? You guessed it, concentration gradient. Now, one thing that's important to note is for every NADH, 3 protons are being pumped across the membrane. Why? Because NADH makes 3 stops along its way to the end. And so it drops off those 2 electrons. Well, NADH doesn't make the stops, the electrons do. And so the electrons stop here, 1, here, 2, and here, 3. And each time they make a stop, they are pumping 1 proton across the membrane. So, a latecomer to the party is FADH2. FADH2 doesn't stop at the first stop. He stops at the second stop, but he still drops off his electrons. And these electrons are going to do the same thing that the electrons from NADH did, except they're not going to power 3 pumps, but only 2 pumps. And it turns back to FAD and goes back to the Krebs cycle to pick up more electrons. And again, these proton pumps are pumping protons into the inner membrane space in order to build a concentration gradient. Now, what happens to these 2 electrons at the end of the road? Who likes used up electrons? Well, there's one molecule that likes electrons more than anything in the entire world, and that is oxygen. Oxygen loves electrons and is always willing to accept electrons. And in order to accept electrons, there also must be the addition of 2 hydrogen ions. And if you'll remember, all the way back to photosynthesis, when this trio was separated by a beam of light, well, they are reunited here again to form H2O. So these electrons go through the electron transport chain. Their energy is used up, and they combine with oxygen. Oxygen is the final electron acceptor. Without oxygen, cellular respiration does not occur, because without oxygen, these electron carriers have no place to drop off their electrons. This is why fermentation happens when there's no electrons or no oxygen, because these electron carriers need a place to drop off their electrons. That's why fermentation occurs. And so when oxygen is present, those electrons are dropped off into the oxygen molecule or the oxygen atom, and that makes H2O. And so when a person is deprived of oxygen, parts of their body begin shutting off because they are unable to make ATP and thus are unable to have life and have energy. And so that is why oxygen is necessary. And H2O, consequently, the reason why when you breathe on a mirror, there is fog there, or water on the mirror, is because H2O is in your breath along with CO2 because of this process. So we have built up a concentration gradient, and our friend ATP synthase knows exactly what to do with a concentration gradient. So large concentration gradient of protons in the inner membrane space, and they will diffuse across the membrane using ATP synthase. And when they do that, ATP synthase is going to spin around. And remember that spinning motion causes mechanical energy, and that mechanical energy is used to create an ATP from an ADP. For every 1 proton that is pumped across the membrane, 1 ATP is made. And so NADH, which pumps 3 protons across the membrane, is responsible for making 3 ATPs. FADH2, which pumps 2 protons across the membrane, is responsible for making 2 ATPs. So let's get to some cellular respiration totals as far as ATP is concerned. Well, back in glycolysis, we netted 2 ATP. And we also created 2 NADPH, or NADH, which translates to, remember, 1 NADH equals 3 ATP, so 2 NADHs is going to equal 6 ATP. So glycolysis gives us a total of 8 ATP. Well, what about when we add Krebs and ETC? Now here's where we're going to get a lot of energy. Well, 2 ATPs were created in the Krebs cycle, just straight ATPs. We also made 8 NADHs, remember? 4 for each pyruvate and 2 pyruvates for each glucose, so 8 for each glucose, which is going to give us a total of 24 ATP. NADH is really chipping in and doing its part. Well, 2 FADH2s also do their part and provide 4 ATP. So what is the grand total? Well, 8 from glycolysis, an additional 30 from the Krebs and the ETC give us 38 ATP. Now, many textbooks will say around 36. And why do they say 36? Well, they'll say that the 2 NADHs that were made in glycolysis require ATP to get across the membrane for to get across the membrane of the mitochondria in order to go to the electron transport chain. So 36 to 38 is the commonly accepted totals for the amount of ATP that is created in cellular respiration. So we've started with glycolysis. We started with a glucose molecule, and in glycolysis, that glucose molecule was broken down into 2 pyruvates. So we started with glucose, we end up with 2 pyruvates. Those 2 pyruvates then are fed into the Krebs cycle one at a time, and they make a total of 8 NADH, 2 FADH2s, which are electron carriers. The purpose of the Krebs cycle, pyruvate feeds in and electron carriers come out. We also happen to make 2 ATPs there as well. And then lastly, the electron transport chain converts each one of those electron carriers with the process of creating a concentration gradient in order to use ATP, and in the presence of oxygen in order to make from those electron carriers ATP.